With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Okay, the question says all of the following are the properties of the enzyme Taq polymerase except, okay. Now, where the Taq polymerase is used, okay, Taq polymerase is actually used in our PCR, that is our polymerase chain reaction. Now, what happens in PCR? In early PCR, there was a problem. What the problem was that they needed to add DNA polymerase in every cycle. Now, what happened? This adding to DNA polymerase in every cycle, it was not just tedious, but also there was a chances of errors. Now, what happened? There was a modification, Mullis and that carry Mullis and his colleagues. Now, what they did, they actually isolated a particular thermostable, okay, thermostable DNA polymerase. So, they isolated a particular thermostable DNA polymerase, which was popularly known as its Taq polymerase. Okay, this is popularly known as Taq polymerase. It's a heat resistant enzyme. It is actually a heat resistant enzyme. It is actually a heat resistant enzyme isolated from the bacteria called the Thermus aquaticus. It is isolated. It is isolated from Thermus aquaticus. Okay, isolated from Thermus aquaticus. Now eliminated, which happened to be a very blessing, or we can say a major achievement for the scientists, because after this DNA polymerase was isolated, there was no need to add the DNA polymerase in every cycle. That is, Taq polymerase helped reduce the process to a much lesser time, and there was no more complications. There were chances of less kind of errors. So Taq polymerase, it is said that it is actually obtained from a thermostable bacteria called the Thermus aquaticus. Now, Thermus aquaticus can survive a temperature of almost 95 degrees Celsius. Now, let us see the options, what it is said. It is a thermostable DNA polymerase, right? Obviously, it is a thermostable. That's the reason it can tolerate such a temperature. It is isolated from bacterium called the Thermus aquaticus. Yes, this is also right. It is used in the amplification of gene of interest using PCR. Yes. It is a thermostable RNA. No, it will not be RNA polymerase. It will be DNA polymerase. So it's a wrong option. So our correct answer will be the fourth one. That is, answer is our 4.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.